I have nothing to disclose. So the focus of our research is on benign prostatic obstruction and its effect on the lower urinary tract system. And to assess this, we use cold cup biopsies from the bladder dome taken from urodynamically defined specific phenotypes. We performed mRNA and microRNA next-generation sequencing on these biopsies, and we looked specifically at detrusor overactivity. And we found that 48 mRNAs are regulated in detrusor overactive patients only, and some of which might be contributing to the urodynamic phenotype we found. So we performed a pathway analysis on these patients with detrusor overactivity only mRNAs. Interestingly, pathwork analysis showed that most of these pathways had to do with contractility, which was not surprising when you're looking at detrusor overactivity. We then performed PCR to validate these findings, and we could show that contractile markers are upregulated in patients with detrusor overactivity. We then looked at adrenergic receptors and phospholamban, and we found that beta-3 and alpha-2 adrenergic receptors were downregulated, whereas phospholamban was upregulated in the patients with detrusor overactivity. We took a quick look at the location of the beta-3 adrenergic receptors, and we could only find these in smooth muscle tissue. We could not find them in smooth muscle cell culture, urothelial cell culture, or urothelial tissue. We then looked at which microRNAs target our findings. So 374A5P targets phospholamban, 2125P targets our beta-3 adrenergic receptor, and 421 targets the 2A adrenergic receptor. Looking at these in our different phenotypes, we found that the 2125P is upregulated in all of them, whereas 374 and 421 are specific. 374 is downregulated in DO only, and 421 is upregulated. So 374A5P is especially interesting because 23 out of the 48 detrusor overactivity only regulated RMAs are affected by this. It is also the targets are also upregulated in our detrusor overactivity data set, matching its downregulation. We then, again, validated our findings looking at microRNA and detrusor overactivity patients, and we actually found that 374A5P is still downregulated in a larger number of patients. So downregulation of the mere 374A5P might have a consequence of gene expression in detrusor overactivity. So in summary, beta-3 adrenergic receptor was downregulated and phospholamban was upregulated in smooth muscle from patients with detrusor overactivity. The mechanism may be that a downregulation of the beta-3 adrenergic receptor increases dephosphorylated phospholamban, which might lead to a calcium overload, upregulation of miR-2125P, and decreases its target beta-3 adrenergic receptor, which then induces a feed-forward loop continuing the cycle. This is the group that did the work. Thank you very much.
Now open for questions.
One of the issues when the bladder becomes overactive is that the cell population changes. So the number of smooth muscle cells often declines. There are more interstitial cells, more fibroblasts. So do you know which cells that you're actually sampling? Because if the population changes, then you're going to sort of get a mixture of transcriptome from all different things, aren't you?
Well, that's the main problem with the cold cup biopsies, and we know that, but we worked with the whole biopsy. So yeah, we cannot tell exactly which cells we're looking at. We're just looking at the wall biopsies.
Right. And is it detrusor only, or do you take off the mucosa, or is it just a single cold cup biopsy?
It's a cold cup biopsy, and it has urothelium and muscle.
Right. Okay. Yeah. Okay, so can you look for specific cell markers, for example, to see if they're up or down?
We did look at specific cell markers to normalize it, and there was not such a big difference.
Right, okay. All right, thank you.
One other question. Did you look at any changes in actual intracellular calcium concentrations in your samples, such as changes in calpain, phospholipase A2, or other things that react to increases or decreases to intracellular calcium itself?
No, we haven't looked at that yet. This is really preliminary data now. It's pretty
That'd be a very good thing to
Yeah.
look at.
Thank
Thank you. No other question? Thank you very much.